Hi, my name is Danish Wafiq.
Hi, my name is Amirah.
Hi, I'm Syaheerah.
Hi, I am Calvin.
I'm Yani. This video is about cancer genome sequencing.
Cancer is the uncontrolled growth of abnormal cells in the body that develops when the body's normal control mechanism stops working. The old cells do not die and instead grow out of control, forming new abnormal cells. These extra cells may form a mass of tissue called a tumor.
Here's an analogy. Just like a piano and its key, when the keys harmonize and play chords together, they can lead to a beautiful piece of music. Just like an organism or a life, a single cell can lead to the disruption of the whole organism, as we can see in diseases like cancer.
There are various types of cancer. There is a breast cancer, thyroid, testicular, prostate, melanoma, cervical, and the list goes on. But no worries, there are treatments for cancer.
The three main treatments for cancer are surgery, where tumors are directly removed. Surgeon makes an incision, finds the cancer, and removes it. Chemotherapy, using chemicals to kill the cancer cells, which can be taken by pills or given through an IV into the vein. Radiation therapy, using X-rays to kill cancer cells.
There is no cure for cancer yet. However, detecting and treating the disease at an early stage can significantly improve a person's outlook. Curing the cancer would mean completely eradicating the condition without it coming back. Although this can happen for some people, there is currently no specific cure for cancer. Some types of cancer have the highest of 5-year relative survival rates. These survival rates are primarily for cancers that doctors have diagnosed at an early stage.
Why is there no cure for cancer? Although different cancers have similar characteristics, but they are very different from one another. Even cancer itself is not homogeneous. There may be 3 or 4 or 5 slight variants in the cancer cells. This explains why it is hard to fully get rid of cancer, because its cancer cells may have different subtypes that will require different treatments.
Now to the fun part. There is now an innovative new diagnostic tool, the Genomic tumor assessment, which examines a patient's tumor genetically by sequencing the cancer cell to identify the mechanism that caused the cancer.
Cancer genomics is basically the study of DNA sequence and gene expressions that differs between the tumor cells and host cells. Let's look at how Dr. George Vasmatzis, the co-director of the Biomaker Discovery Program within the Center for Individualized Medicines, differentiates between the gene sequence of a normal cells and a tumor cells.
In cancer cells, the information is missing. This is a genome of a cancer patient. All the chromosomes are represented here. Chromosome 1 on top in this box, and when you have a little gray dot, that means you have that part of the information correct. But if you have red dots or blue dots, something is wrong. This is a normal genome. So this cancer genome on chromosome 6 has a big deletion. That means a lot of those cells on chromosome 6 are just gone. Or in that case, back there, you have exchange of information between chromosomes 2 and 16 in that case. It's like taking little paragraphs or books from one chromosome and put them on the other. You start with a romance story and you end up with a zombie story. So all this is not good.
A major goal of cancer genome sequencing is to identify driver mutations, genetic changes which increase the mutation rate in the cell, leading to more rapid tumor evolution and metastasis.
Sequencing DNA involves determining the precise order of the 4 chemical building blocks or bases that make up the DNA molecule. The bases, which are A, C, P, and G, spell out the genetic code for each cell and each organism. Knowing the sequence of bases, also called nucleotides, helps scientists understand how different segments of DNA function.
This may direct to tailor treatments in very specific ways. Unlike chemotherapy, it acts like an atomic bomb that destroys everything. Whereas genomic sequencing as a cancer treatment, it works just like drone attacks where the targets in the genes are more specific.
How is this done? Let's have a look at how Dr. Nicholas E. Nevin, an assistant professor at the University of Texas, sequenced a single cancer cell.
So this is our cryomicrotome system, which allows us to keep the tissue frozen while Anna goes through and cuts it with a very, very fine blade that will cut sections that are only 1 cell thick throughout the tissue. We're going to be looking at an early stage breast cancer called ductal carcinoma in situ. And we'll see ducts at the earliest stages of tumor development where the tumor cells have started to proliferate within the ducts. Anna is going to use this 1 micron UV laser to cut around a cell and then use another laser to burst that cell out of the tissue and into a collection vessel. So now we're going to go through the whole genome amplification process to amplify the DNA from 6 picograms up to nanogram amounts so that we can generate sequencing libraries and do our quality control steps. So all of that work has to be done here under a clean hood, which is sterile, and it's very critical before the DNA has been amplified not to contaminate it with any bacteria or even viral DNA. Okay, so this is the 3rd step of single-cell sequencing. It's the next generation sequencing part. So this is one of the runs that's going on right now on the Illumina machine. If we sequence the whole genome of a single cell, we'll be getting back about 1 terabyte of data from each cell. It's really important for informing precision medicine or personalized medicine and guiding targeted therapy. Try and get these tools into the clinic where they can make a big impact on reducing morbidity. And that's why getting that single-cell genomic information is so important for understanding cancer.